Hello, I'm Damla Senol Cali. Today I will be presenting our work GenASM, a low-power memory-efficient approximate string matching acceleration framework for genome sequence analysis, and this work will appear in Micro 2020. Let's start with some background. The genetic material of an organism is called genome, and each genome consists of DNA. And the information in DNA is encoded with a sequence of 4 different types of bases, A, C, G, and T. And the genome sequencing is the process in order to determine the order of the DNA sequence in an organism's genome. And why do we care it? Because it is pivotal in personalized medicine, outbreak tracing like COVID-19, evolution, and forensics. Due to the limitations of sequencing devices, the whole genome of most organisms cannot be sequenced all at once. So first, the genome is broken into smaller fragments, and each of these smaller fragments are sequenced, and we obtain small pieces of DNA sequences, which are called reads. And these reads lose their information with respect to the original, like location information with respect to the original genome. So that's why genome sequence analysis requires taking these small DNA fragments and then reorganizing them into the entire genome. And the success of all medical and genetic applications critically depends on the existence of computational techniques which will process and analyze this enormous amount of sequence data, genomics data, quickly and accurately. However, effectively leveraging genome sequencing as a tool, as expected, requires very high computational power, and we have to process a large amount of data. So it is bottlenecked by the current capabilities of the computer systems. A common approach to genome sequencing is to perform read mapping. And in read mapping, we assemble the reads by finding their location in a reference genome. And we are aligning the reads against the reference genome in order to detect the matches and variations. And this is one of the key components of genome sequence analysis. Unfortunately, the bases in a read may not be identical to the bases in the reference genome at that original location for one of the 2 reasons. First, the sequencing technologies introduce errors into the read fragments during extraction. And second, the read may contain mutations that are specific to the individual organism that the DNA was extracted from, which are not captured in the reference genome. So when we consider reference as a text and read as a pattern, we can observe 3 different kinds of errors. The first, we can see a deletion. So the character doesn't appear in the pattern, but it was existing in the reference. Or one of the characters or some of the characters may flip, so substitutions may occur. And also a character that doesn't appear in the text, like reference, can appear in the red, which is called insertions. And multiple steps of read mapping must account for these errors. Due to its need to account for edits, multiple steps of read mapping is effectively a form of approximate fuzzy string matching, and can make use of existing approximate string matching algorithms. Typically, approximate string matching uses an expensive quadratic time dynamic programming algorithm, and this algorithm accounts for over 70% of the execution time of read mapping and acts as the bottleneck for the entire genome analysis pipeline. And because of that, in this work, our goal is to accelerate approximate string matching by designing a fast and flexible framework so that we can accelerate multiple steps of the genome sequence analysis pipeline. Let's look at the approximate string matching algorithm we focused in this work. So we have focused on the Bitap algorithm, and in order to replace this expensive dynamic programming algorithm. And the reason is that this algorithm can perform ASM with fast and simple bitwise operations, which makes it amenable to acceleration. And there are 2 common steps of the algorithm. The first one is pre-processing. So for each possible character in the alphabet, and in this context it is ACGT, we are generating a pattern bitmask, which indicates if that character exists at each position of the pattern. And the second step, searching or edit distance calculation, is the main body of this algorithm. And we are comparing all characters of the text with the pattern by using these pre-processed pattern bitmasks a set of other bit vectors that hold the status of partial matches, and we use only bitwise operations in order to perform this calculation. So each bit vector has a length equal to the length of the pattern, and the semantics are reversed. So 0 means match, 1 means mismatch. So let's look at the first step, pre-processing, and assume that this is our pattern, and we need to generate those 4 pattern bitmaps. So starting from the leftmost character, we are iterating over the given pattern. And if we see the corresponding letter, which is A in this context, we put a 0. For all the other positions, we have 1. And we can fill the rest accordingly. So when we look at the second main step, searching, I won't go into the details due to time limit, but I will highlight key properties of the algorithm. And for more details, you can refer to our paper. So first, we are dealing with large number of iterations. Second, there are data dependency between these large number of iterations, so there's no room for parallelization. But the promising part is, as we highlighted, we are only dealing with very simple bitwise operations. So these data dependencies limits the efficiency and the scalability of the algorithms on CPUs and GPUs. And although we can obtain text level parallelism by dividing the text into multiple chunks and searching them in parallel, we are limited by the number of compute units in existing systems like CPUs. And also there's a high memory bandwidth requirement to read and write the computed bit vectors. So we revealed that both CPU and GPU systems are imbalanced for this algorithm. And there are 2 additional algorithmic limitations of the baseline Bitap algorithm. First, there's no support for traceback, which is a critical step of read mapping. That this step finds the sequence of matches, substitutions, insertions, and deletions along with their position. So it's not finding the location, it's also finding the actual alignment information as well. And also there's no efficient support for both short and long reads, because as I mentioned, the bit vectors have the length equal to the pattern length, which is the read length. So that's why we have to divide the bit vectors into word sizes of the machines. So it is not practical. Because of these limitations, in this work, we propose GenASM. It's an approximate string matching acceleration framework based on the Bitap algorithm, but we optimize the algorithm to fix the limitations it has, and also we propose a new hardware design for the modified algorithm. And we obtain a highly parallel Bitap algorithm with very small memory footprint. And we also develop a novel bit vector based algorithm in order to perform traceback on top of the Bitap algorithm. And we design processing in memory accelerator for both components, Bitap and traceback. And we propose a fast, efficient, and flexible framework which can accelerate multiple steps of the genome sequence analysis pipeline. And we optimize our design for 2 types of sequencing technology data. So one of them is short yet accurate, and the second one is long but noisy read. So both of them are existing. So that's why our goal is to optimize our design for both scenarios. So we modify the baseline Bitap algorithm for enabling efficient alignment of longer patterns, so longer reads, and removing the data dependency between the iterations and providing parallelism, and also providing support for traceback. And both our Bitap algorithm, the modified algorithm, and the new traceback algorithm, since they are representing the query reads as bit vectors, we are also taking advantage of bit parallelism during the computation. And our traceback algorithm provides both full support for edit distance calculation, where the errors have unit costs, and also minimal support for non-unit costs for edits, and more complex scoring schemes, which is also important. So when we look at our design, so GenASM is composed of two main components, GenASM DC and GenASM TB. GenASM DC generates bit vectors and performs edit distance calculation. So DC is coming from distance calculation, whereas GenASM TB performs tracebacks and assembles the optimal alignment. So TB comes from traceback. And we also have DC SRAM and TB SRAM. So DC SRAM is for writing the intermediate data required for DC execution, and TB SRAMs are for writing the bit vectors generating during DC execution, which will be later used in the TB execution. So in order, DC and TB accelerators are tightly coupled with each other during execution, so we have to put them near each other, and we can port our accelerators anywhere in the system. However, in order to achieve high parallelism and low power consumption, we place both of our accelerators in the logic layer of the 3D stack memory. And that's why we achieve high parallelism and low power consumption. So let's look at the overall mechanism of GenASM. So when read mapping starts, the host CPU generates candidate locations in the reference genome for each reference, for each read fragment. And then, excuse me, for each candidate location, GenASM DC reads the corresponding reference region and the read from the memory and write these into its dedicated SRAM, which is DC SRAM. And after that, DC searches for the pattern within the text region using the modified Bitap algorithm and generates the bit vectors. And each processing element of DC writes these generated bit vectors to its own dedicated SRAM, which we call TB SRAM, as I mentioned. And once DC completes its search, then TB starts reading the stored bit vectors from TB SRAMs and generates the traceback output. And once GenASM TB generates this output, it moves on to the next reference read pair. So in order to reduce memory bandwidth and memory footprint, we implement GenASM DC accelerator as a linear cyclic systolic array. And processing core is the basic compute component with capability to compute the intermediate bit vectors for different error types. And it's a very basic logic. And we add the required logic around a processing core to define a processing element, and multiple processing elements are concatenated with appropriate data multiplexing in order to define processing block. And similarly, also TB hardware design is pretty simple, and it only requires to achieve three steps. The first, it is reading the bit vectors from one of the TB SRAMs using the computed address. And then it performs a very, again, basic, simple bitwise operations for computation and comparison. And this helps us to find the traceback output for the current position. And by using these comparisons and computation, we are also computing the next TB SRAM address to read the new set of bit vectors. And then we go back to step one. And after GenASM TB finds the complete traceback output, it writes the output to main memory and completes its execution. So we design our system around the 16 gigabytes of 3D stacked hybrid memory cube, so HMC memory with 32 volts. And within each volt, the logic layer contains a GenASM DC accelerator, its associated DC SRAM, a GenASM TB accelerator, and a TB SRAM. So by doing this, we are achieving volt level parallelism because each accelerator at each volt can work independently. And we divide the reference text into like subtext and store each subtext in different volt like memory, like I'm showing here. And we search the query at each volt in parallel so that this helps us to achieve text level parallelism as well. So in order to understand what are the use cases of GenASM, we need to look at the read mapping pipeline. Read mapping can be seen as a four-step process, which is also called seed and extend. First, a typical read mapper starts with indexing the reference genome. And this index contains all possible fixed length substrings as the keys and the exact match locations of these substrings in the reference genome as values. Second, seeding queries the index structure for determining potential mapping locations of each read in the reference genome using seeds like substrings from each read. This provides the potential mapping locations. So before going to the last alignment step, if you want to further eliminate some of these potential mapping locations, we have the third step, pre-alignment filtering, which uses filtering heuristics to examine the similarity between every read and its potential matching segment on the reference genome that are identified from the seeding step. And these filtering heuristics aim to eliminate most of the dissimilar sequences to decrease the number of required alignments. So the last step, read alignment, also called seed extension, for all the non-filtered candidate mapping locations, read alignment step performs a dynamic programming-based alignment to determine the best location of the query read. And as part of this step, traceback operation between the reference segment and the query is also performed in order to find the optimal alignment. So the steps that include approximate string matching is our use case, our use cases, and here indexing, pre-alignment filtering, read alignment, so all of the steps except seeding include ASM. So that's why we have three main use cases which we also evaluated as part of this work. So first, read alignment, so it can perform ASM between the query reads and the candidate regions and report the optimal alignment. And for the second use case, pre-alignment filtering for short reads, which requires finding the dissimilarity between sequences, GenASM can efficiently calculate this similarity, dissimilarity, and perform the filtering. And the third use case is a fundamental operation in genomics, which is edit distance calculation, which is measuring the similarity distance between two sequences. And GenASM DC, as we explained, is inherently an edit distance calculation accelerator, so it's another natural use case of GenASM. We also discuss other possible use cases of GenASM in our paper, and one of them is generic text search, for example. For more details, please refer to our paper. So let's move on to the evaluation. And as I explained, we use 3D stacked DRAM architecture to model our design. And we have two different data sets to show our applicability for both short and long reads. And I will show the details of the evaluation of the first use case, which is read alignment, as part of this presentation due to time limitation. And we compare GenASM against state-of-the-art software and hardware for this use case. When we look at the area and power results of GenASM, so when both accelerators operate at 1 gigahertz, and when we synthesize our accelerators using Synopsys Design compiler, we obtain that total power consumption for all 32 volts, which means 32 accelerators along with their associated SRAMs. We need 3.24 watts of power consumption, and the area overhead, again, for all 32 volts is 10.69 millimeters square. When we look at the results for the long read data sets, like for read alignment, we observe that for the software baseline Minimap2, we obtain 116x speed up and 37x less power consumption. And compared to other software BWAMM, we obtain 648x speed up and 34x less power consumption. And compared to state-of-the-art hardware for long read alignment, which is Darwin's GACT, we obtain 3.8x better throughput while achieving 2.7x less power consumption. And our trend is similar for short read. So again, compared to Minimap2 and BWAMM, we observe significant amount of speed up and power and significant power efficiency. And compared to the state-of-the-art short read hardware alignment accelerator, which is ScyllaX of GenX, we observe 1.9x better throughput, and then we have, so GenASM and ScyllaX have comparable area and power consumption. So a very quick overview for the other two use cases that we have evaluated. For the second one, pre-alignment filtering for short reads, compared to the state-of-the-art FPGA-based filter, SUJI, we observed 3.6x speed up, and also GenASM significantly improving the filtering accuracy on top of the speed up. And for the third use case, edit distance calculation, compared to a software library, EDLib, for different size data sets, we observe around 250x to more than 5,000x speed up. And you can see our Micro 2020 paper for more details. So in this work, we tackle the problem of approximate string matching, because it is one of the key bottlenecks in genome sequence analysis. And our goal is to provide an ASM acceleration framework in order to accelerate multiple steps of genome sequence analysis. And our key contributions are: we present the first work to enhance and accelerate Bitap algorithm for ASM with genomic sequences. And GenASM is the acceleration framework, where we co-design our modified, scalable, and memory-efficient algorithms with low power and area-efficient hardware accelerators. And we evaluated 3 different use cases of ASM in genomics. And we show that GenASM is significantly more efficient for all the 3 use cases in terms of throughput and throughput per unit power than state-of-the-art hardware and software baselines. Thank you.